Zebrafish are a common model system for studying developmental biology because they grow very quickly, so you can do a lot of experiments in a short period of time. The unique aspect of zebrafish is if you cut off a large piece of their heart, they can actually regrow new heart muscle. Human hearts undergo some type of injury, like a heart attack. New muscle tissue cannot grow back. Instead, there's just a scar that remains.
So for us, we would really like to study zebrafish specifically because they're able to regrow their hearts. And hopefully we're able to isolate different factors that encourage this process and then really see if we could translate them to human hearts.
The problem is, if you want to study the actual heart regeneration process, the conventional approach is to take a heart out of the zebrafish and take images at different time points and just really get a snapshot view of the regeneration process. So in conventional culture conditions, zebrafish hearts last maybe 3 to 5 days. So we partnered up with Ellen Lean's lab at Children's Hospital Los Angeles to see if my lab could fabricate a fluidic device that could house zebrafish hearts taken out of the animal and keep the hearts alive, and then be able to study them for longer periods of time outside of the fish.
Through a very long iterative process with a lot of rapid prototyping, we were able to taper it down to a device with a footprint that's only 22 by 22 centimeters. Our fluidic device was really able to hold 4 zebrafish hearts at one time and keep them alive to study all the regenerative properties that we're interested in.
So when we culture zebrafish hearts in our fluidic device, after 1 week, the structure and function is actually quite similar to a freshly explanted heart. We have been able to keep hearts alive for up to 2 weeks.
So right now we're imaging the hearts inside of our device, and we can actually see the gap of where the tissue used to be. And over the course of 4 days, we're able to see the blood vessels slowly move in and close the wound site, and then really begin the regenerative process.
So what we can potentially learn from the zebrafish is certain molecules or factors or hormones that could maybe boost the regeneration process in a human heart that's been injured by something like a heart attack.